Welcome everyone to the Fermentation Association's webinar, The Science Behind the Safety of Fermented Vegetables. I'm talking today with Fred Breidt, a noted microbiologist with the Agricultural Research Service Division of the United States Department of Agriculture. Fred's laboratory is based at North Carolina State University in Raleigh. I'm David Ehreth, founder and president of Alexander Valley Gourmet, parent company of Sonoma Brinery, and I'm on the advisory board of the Fermentation Association, a trade group that was launched to support producers who use fermentation to create delicious and often healthful foods and beverages. TFA has the goal of helping to educate consumers about fermentation and its benefits, supporting scientific research into those health benefits, and working with food safety authorities to establish clearer and more appropriate regulations in regards to fermentation. My company produces, among other things, fermented pickles and sauerkraut, so today's session is of particular interest to me. We have a number of questions already submitted for Fred, but please send any additional ones during the course of today's session, and we'll try to get to them. So I'd like to introduce to you Dr. Fred Breidt. If I can share with you a brief anecdote about how I first contacted Fred. I was sitting on top of a couple of tons of sauerkraut that had been infected with yeast and honestly had no clue what to do. So what did I do? I called Ghostbusters. That's Fred Breidt. And Fred was terrifically helpful to me, very responsive. And so let me just tell you who Fred is. Dr. Breidt received a BS in genetics from Ohio State University and a PhD in microbiology from the University of Kansas. He has been with the USDA Agricultural Research Service as a microbiologist since 1990. He is currently a lead scientist at the ARS Food Science Research Unit located in the Food, Bioprocessing, and Nutritional Sciences Department at North Carolina State University in Raleigh. Dr. Breidt's research centered on the study of vegetable fermentations and bacterial competition, including fermentation ecology, mathematical modeling of bacterial competition, buffering and pH, genomics, bioinformatics, and acid resistance of bacterial pathogens. Dr. Breidt has authored over 100 peer-reviewed research publications and book chapters, mostly on fermented and acidified foods. Dr. Breidt is recognized internationally as an authority on vegetable fermentation, microbiology, and processing, and the safety of fermented and acidified foods. So with that, I'd like to present to you somebody that I regard as a guru in our industry, Dr. Breidt, and I think he's going to give us a little overview on what he does and what the ARS does. So Fred, it's all yours.
Well, thank you, David. I appreciate that kind introduction. My plan for today was to share a short slide presentation with you that may cover a lot of the issues that you might have questions on. Given talks like this before to other groups, and the issue of fermentation safety is one that comes up a lot, and people are getting excited about the fermentation world these days. Fermentations are increasing in popularity. So I'm going to share my screen and go with a slide presentation here, if I can. Hopefully you can all see that. So what we've got in terms of a talk today is going to be focused primarily on the science behind fermented vegetables, not so much the things that you need to do for the best quality or the best flavors and things like that. First, I want to tell you a little bit about our laboratory. We are actually the Food Science Market Quality and Handling Laboratory let me go back. There we go. In Raleigh, when we're located at NC State University. I'm going to move my monitor a bit so I'm not staring away from you while I'm talking. So the the Food Science Market Quality and Handling Research Unit has been at NC State in one form or another for quite a long time. We have a database of publications which you can find on our website using this tab here. If you look for us, an easy way to find us is Google Food Science Research, USDA Food Science Research, and we should be the first link there. Alternatively, you can Google my name and USDA and you'll get related pages that can bring you right to this home page. Our laboratory studies things like pickled vegetables, salad dressings, sweet potato processing, and peanut processing. And what you're looking at here is basically the pickle team. This is our research leader, Dr. Mukarab Qureshi, and Dr. Elenis Perez-Diaz is one of our pickle scientists, and Dr. Suzanne Johanningsmeyer is one of our pickle scientists. And then we have a couple support staff, I will go on to the next slide. So we've been there since 1935 or so, or 1932 or 1933. Anyway, the early 1930s. The first publication from our group was in 1938. We have over 400 papers on our website at that link I just showed you. Most of those have PDF links, and they cover different areas of fermentation technology, pasteurization and processing, which was developed originally for pickles in our lab in the 1940s, texture and quality, and food safety. And this paper that you see there is the first paper from 1938 from our group. So I wasn't there then, but not that old. So just very briefly, I know you all have a lot of knowledge of fermentation, or most all of you on the call would, or on the video will have a good knowledge of fermentation technology, but many people argue about where this technology came from. My thinking is at the end of the Pleistocene era, about 10 to 12,000 years ago, when the Ice Age was retreating, primitive pots, and these come from the Korea Straits region between Korea and Japan, which as the Ice Age was retreating was actually becoming impassable. These are ancient clay pots that, you know, if you're going to make a pot, what are you going to do with it? And these vessels could very well have been used for fermenting or for holding vegetables in. If you're a hunter-gatherer and you're going to gather stuff, you know, and you live near the ocean, putting your vegetables into a pot, a little salt water, they'll be there for months and still edible. And if you fast forward to today, you can get to a situation like this. This is a rather large, not the largest, but one of the largest pickle plants in the United States. And this particular plant has over 1,000 tanks of 40,000 liters each, all filled with cucumbers. So that's a big brine yard. And that's some of the technology they use for loading those tanks. So that's 10,000 gallon tanks. And so when we start with the first mention of kimchi by Yi Kubo, and I probably butchered the name, from 1200 AD, we've come a long way. And so here's some, I just thought I'd throw in a slide of modern pickle processing for you. Like I said, we've had a long relationship with the pickle vegetable industry, and there's that. So food safety of fermented vegetables. Aren't they safe? What are the hazards? What are safety factors that you need to pay attention to? And those are some of the main questions that a lot of people have when we talk about this topic. And one of the first things that you need to think about if you're thinking about safety of fermented and acidified vegetables is pH. And pH 4.6 is a critical control for acid and acidified vegetables. And Dr. Keith Ito, who is actually still active and working as in charge of process authority work in California out of UC Davis, published this paper in 1976. He said pH 4.8 would stop Clostridium botulinum from growing, but FDA and others have found that 4.6 is a little bit better mark to shoot for, considering that botulism kills people rather easily, and one case is considered an outbreak. So we use this 4.6 number, and it is a hard stop critical control for acidified foods, which are not fermented, where you add acid to fresh vegetables to bring the pH down below that, and then store it anaerobically. This pH is normally achieved in most all vegetable fermentations due to the presence of sugars, and they're fermented to organic acids, and we'll go over that in a minute. It's not critical that this pH be achieved immediately in your fermentation, and I'll tell you why. As long as the pH is going down and you're getting acid produced while the fermentation is going, you're in good shape. So I wanted to mention about botulism just a little bit. This is a meat fermentation slide, but the word botulism actually comes from the Latin word for sausage, which is botulus, just in case you are interested in making sausages. The reason is in a previous era, a lot of these things caused botulism for people because the conditions were right for Clostridium botulinum, the organism that causes that, to grow. It was anaerobic, there was a high enough water activity, and the pH was above that critical 4.6 limit. One of the key things I mentioned is bacterial competition is a really important control for preventing botulism. In addition to that pH 4.6 mark, the growth of lactic acid bacteria in particular, but other organisms, actually are very inhibitory to the outgrowth of the spores that can germinate and cause botulism when the organism grows. So we'll talk about that a little bit more. There are other organisms as well that I think you need to be somewhat concerned about: E. coli, Salmonella, and Listeria. And of those three, in pickled vegetable products, we know that E. coli, I have allergies, pardon me, I do not have COVID as far as I know. So, E. coli, particularly the pathogenic O157H7 and related serotypes, are very acid resistant, and they can survive for a long, long time, much longer than Salmonella and Listeria, even under cold conditions, because Listeria likes cold temperatures. These organisms can certainly be on and in fresh vegetables, and I will show you a slide of organisms in fresh vegetables. So when I say on and in, I actually mean that. This is the surface of a cucumber micrograph, and what you see is a stomate on the surface of the cucumber, and that's where the cucumber lets gases exchange. When you harvest cucumbers, a lot of farmers will take them and they'll put them in cold water to get the field heat out of the vegetables. And this is so that when they are transported from the farm to wherever they're going, they stay fresh looking. But what that does, going from a hot environment to a cold environment, the gases inside the cucumber contract, and it can literally suck in microorganisms that happen to be on the cucumber or in the water, because you're putting cucumbers into water, that they'll come off that, and then and so you can see that bacteria, just washing the surface isn't always going to do the trick for you. And so these bad guys are the ones that we're concerned with primarily because of their extreme acid resistance, and that's this E. coli bug. It has either anaerobic or aerobic metabolism. It is primarily homofermentative. It'll make lactic acid, a little bit of acetic acid too. It is salt resistant, grows just fine, pretty well, up to almost 6%. And it can survive but not grow once the pH is below 5. Survive is important. The infectious dose, that's a term scientists generally don't use anymore. But the amount of organisms you have to actually eat to get sick is very, very small. People have gotten sick from hot dogs where they found like less than 1 organism per gram in the hot dogs. So it's an organism that we worry about a little bit. It is very much able to survive for long periods, even at the pH values of most acid and acidified foods. And it can survive even better at those pHs at cold temperatures. Where do you find E. coli? Pretty much everywhere you look for it. This is a map of the Salinas Valley in California. And the little red dots that you see on that graph with the circles and arrows basically are showing you when and where E. coli was found at different times of the year and different places in the Salinas Valley River water and on the riverbanks and so forth. This is a paper by one of my ARS colleagues, Michael Cooley, at the Albany lab, USDA Albany lab in California. So it's out there. Can it be a problem in fermented vegetables? And the answer is yes. In Korea, there was a very large outbreak. And down here, you can see estimated 230 school children were put in a hospital from some very lightly fermented kimchi. Many people like kimchi very lightly fermented. If the folks had eaten the cabbage that this kimchi was made out of, they would have gotten sick as well. Was the cabbage improved in the sense of maybe had fewer E. coli on it because of fermentation? Yes, but there were still enough when this was eaten by a lot of people to make a lot of people sick. So it's a real thing. And so some studies have been done in the US, in Korea, and other places. And there's a website that you might want to look at sometime called Barf Blog. And it has a story about maybe that fermentation isn't working so well. 1,642 sickened outbreak of E. coli with school children in Korea in 2012. So the 240 number that I had was, I think, an earlier estimate. But pathogenic E. coli salmonella can survive in kimchi during fermentation. Survival of E. coli salmonella and listeria and staph in kimchi. So sometimes the wheels can fall off if you're not careful. And so the moral of the story is, if the vegetables are safe to eat before you ferment them, they're going to be safe to eat after you ferment them. If they're not, you got to ferment them for a long time to make sure they're safe. And I'll show you some data on exactly how long. So then there's the good guys, the lactic acid bacteria. And this is a picture of Leuconostoc mesenteroides from the Joint Genome Institute in California from the original sequencing of that organism many years ago. These organisms, all the lactic acid bacteria in general, are very salt resistant, can grow up to even 10% salt. And there are organisms that will grow even higher. Some pepper mashes are fermented at 15% salt or greater. And they usually have slightly different microbiota than you would get in a typical vegetable fermentation. But they do ferment and they do produce acid from sugar. Most lactic acid bacteria can grow down to even pH 3.3. The Leuconostocs stop a little earlier in the process in the upper pH 3 range. And these bacteria, they're anaerobic. They don't need oxygen. In fact, oxygen is toxic to them. And they actually use manganese ions from vegetable material to help survive the toxicity of oxygen. So I'm not going to go through the details of the biochemistry, but I will point to paper number 283 on our website, which has this lovely graph of the major fermentation pathways if you are interested. And most of the references that I show you, I will include a number with a hash mark on it. And that number is the paper number in our pickle bibliography. By the way, if you Google pickle bibliography, you'll get an older version of our most current database. If you go to the website I showed you at the beginning of the talk and the publication link, you'll get the most up-to-date version. And the PDF is there. You can click on it and download the paper. So in vegetable fermentations, you're basically taking glucose or fructose and making lactic or acetic acid. That's the main story. Cucumbers have a lot of sugar, about equal amounts of glucose and fructose. And these are free sugars. So if you hate fructose, don't eat vegetables. Cabbage is the same way. It's got about 4 to 6%. And what happens is, as the lactic acid bacteria grow, they make acid, lower pH. And this is a graph of competition between Leuconostoc mesenteroides and E. coli. And you see the E. coli growing and then dying. But if you grow them in separate tubes, this is mixed together, they both grow to about the same extent. And this is in a 2% salt cucumber juice brine, which should be very similar to many types of vegetable fermentations. So a point I want to make about salt effects. I've seen some of the questions that folks were interested in, and this is one of them. Salt does not so much inhibit the pathogens as it aids the lactic acid bacteria. And these graphs basically show you the growth rates, the rate at which the cells are dividing for a cocktail of E. coli strains as well as a cocktail of lactic acid bacteria. And the bars are pretty similar for most of the range of different salt conditions we tested. And when you get up around the really high salt concentrations of 6% or so, you start seeing some significant differences, but you can't rely on salt for safety is the point. It does encourage the lactic acid bacteria and it helps them grow, and it will increase overall the safety of your fermentation. Another point that I think is important for folks to know is how you cut the cabbage. So this is actually a type of kimchi, and how you cut the vegetables. And this is a nabat kimchi, which is radish included. And it's a watery type of kimchi. But how you actually cut the vegetables can make a big difference in the microbiota and the quickness at which the red bars sort of fade away. And the red bars are the enteric bacteria, which would include things like E. coli. And so at different temperatures, the higher the temperatures, the bluish and blue-greenish colors are the lactic acid bacteria, are going to do well as the time goes on. And the reds and oranges, the ones we don't want, are going to die off. But you can see that there are significant effects between cut and uncut. And that's because the sugar comes out more quickly into the brine. I got a few more slides, and then I'll take questions. But in terms of pH, we've been able to do some research with a variety of different kinds of commercial fermentation brines and looked at what pH is needed to achieve safety, if you will. And this is safety in terms of what's marked on the y-axis called a 5-log reduction time in days. 5-log reduction means 100,000-fold reduction in the number of E. coli, 100,000 times reduced. And so when you start out at a high pH, that can take several weeks. When you get to a lower pH, just a few days. But it's not zero. Even down at 3.3 or so, it can take 3 or 4 days for that kind of reduction of E. coli. And this is the standard that FDA uses for commercial safety practices for under the Food Safety Modernization Act, which fermented foods are covered under. And so pH 4.6 is a critical value for botulism. But what we're talking about here is those other pathogens that could get on your vegetables. If you source your vegetables and they're clean and they don't have E. coli on them, you're good. But you still need to worry about botulism, but you're good in terms of these other pathogens. But it's very difficult to know that. And so commercial producers and others can rely on data like this based on an easily measurable variable pH. And there's one other kind of thing that I think fermentation people, particularly the common types of vegetable fermentations, kimchi, cabbage, sauerkraut, pickles, need to know about. That's secondary fermentation, which can happen. And in these types of fermentations, the lactic acid can disappear, and you can get propionic acid and butyric acid produced. You'll know it if this happens. It smells terrible. And the pH will go up significantly over 4.6, bringing you into that botulism danger zone. These fermentations are fairly rare, but they can happen with low salt vegetable fermentations. And I use that term to be about 2 to 3% as low salt. Commercial pickles, for example, are usually fermented around 6% because it gives you a very nice, firm, superior quality. They wash that salt out before they put it in the jars to sell it to you, or before you eat it on your hamburger at McDonald's or fast food kind of place. So this can happen. It's easy to detect when it does because it will smell bad, but you can actually get botulism forming before that happens. And molds can cause pH to rise too. We'll talk a little bit about that as well. But molds causing pH to go up, when you get film yeast and stuff on the top of your fermentation, that's aerobic yeasts and molds growing. They can cause this kind of thing to happen as well if they grow to a large extent. And many people say, oh, just scrape the mold off, it'll be fine. And almost always that's going to be true. But what I would almost always say as well is keep a close eye on the pH. And you can do this even with pH test paper, you know, people use for fish tanks or something. If it has the range that will allow you to see pH 4.6, you want to be below that. And that's a simple and easy way to make sure you don't come into a situation like this and end up with some problems. And this is an organism we actually isolated. We called it Pectinatus sodacetanus, which is kind of an Italian Latin word for pickles. And it was one of the organisms that was causing that, or helping cause that spoilage fermentation. That was kind of fun. So this is my basically last slide, some take-home messages. The things that are important in vegetable fermentations are bacterial competition, an anaerobic environment, acid production, and pH. These are the keys to safety for fermented foods. The bacterial competition, as the organisms are growing, will help prevent the outgrowth of Clostridium botulinum even before the pH drops below 4.6. And I can tell you that we know this is true because in every one of you watching this, your upper intestinal tract is anaerobic. Right after, right downstream from the stomach, it's anaerobic. It has a high pH. The pH is usually above 4.6. There's plenty of water, nutrients, and food, and the water activity is high. All the conditions needed for the growth of Clostridium botulinum. Yet we eat those spores all the time in food because they're everywhere in the environment. We don't all get sick from botulism, and it's because the other microorganisms growing in your intestinal tract are keeping those organisms from germinating and growing. They're very poor competitors. So you may have heard, don't feed honey to babies. The reason is honey can have those spores on them. The intestinal tract of babies is not very well developed, and you can get infant botulism through feeding them too much of the wrong kinds of foods. Anyway, the next point, salt primarily aids lactic acid bacteria. I abbreviate them LAB. They may not do a lot to inhibit the pathogens directly, though. Fermentation typically improves the safety, but the quality of the vegetables you're using is critically important. A steady decline in pH and then no increase above 4.6 is really important. And that decline in pH doesn't have to happen overnight. If you have cold temperatures, it can take a long time for that pH. As long as it's steadily dropping and that competition is there, you're going to be in pretty good shape. Then finally, molds can raise pH, but this is easily checked even with pH paper. And tasting is not a good idea because tiny, tiny amounts of botulism toxin can kill you. Even by dipping your finger in something and licking it can be a lethal dose. So I want to say one more thing, and this is, I wasn't planning on talking about this, but very recently, a new paper came out in a systematics journal on the taxonomy of lactic acid bacteria that anybody interested in vegetable fermentation might want to know about. The genus Lactobacillus was getting very, very bloated. There were over 261 different species of lactobacillus. And this was becoming a problem, and taxonomists, primarily in Europe, took the tiger by the tail and tackled this problem using molecular methods to look at whole genome sequences and other things. And there is no longer Lactobacillus plantarum. You may be surprised to hear that. It is now Lactoplantibacillus plantarum, subspecies plantarum. So that is what we have now from the taxonomist world. And if you try to publish a paper, a research paper today in food science, and you're talking about lactic acid bacteria, and you don't use this new taxonomy, your paper's not going to fly. So it's they're enforcing this. So we are now Lactoplantibacillus plantarum, if you're interested. And the paper can well, I guess these slides will be made available. You can track it down if you want to look at it. It's freely available on the Microbiology Society Journal website. So that's my presentation. I always show this slide at the end of talks, or a similar slide. And what this shows is all the vegetables, and behind it you see this slightly creepy looking smiley face. That's the microbial world. And the microbial world is smiling because most all the bacteria in the microbial world are not harmful to you and are often very helpful. But there's a few, so few that you can't even see them around the edges of the microbial world that are harmful, but we do have to pay attention to them because they can have severe consequences. There's my email address. I'm always open to take questions. I work for you all. You all pay taxes. I get paid by that. Actually, I pay taxes. They pay me and they take it back. I never understood that. But in any case, feel free to drop me a line if you've got a question or need some help. And so with that, I will stop sharing my screen and go back to the moderator, David.
All right. Well, thank you, Fred. That was really fascinating. If I could indulge myself in a question for you, there's been some discussion on my end. This is news to me about Lactobacillus plantarum, so I'm going to have to internalize that, and I'm going to go find your taxonomy. One of the things that we were sort of noodling over was the subspeciation of Lactobacillus plantarum, because in the old days, I guess, when we used to call it Lactobacillus plantarum, we were aware, if you go to the ATCC website and you look at all the different subspecies of plantarum, if any work has been done to isolate subspecies, and I guess now since we've subdivided the category even further, has any work been done that you're aware of that, like in the wine industry where they sell yeast, and the yeast, it's all Saccharomyces cerevisiae, but this one sugar tolerant, that one's alcohol tolerant, so on and so forth, and they have different, different mouth feels and all this. Has anyone done any work on bacterial subspeciation with regard to the organoleptic, characteristics of their metabolites?
Yeah, I can tell you that there's work on this going on. In fact, my colleagues, Dr. Elenis Perez-Diaz and Dr. Suzanne Johanningsmeyer, who will be giving talks in this series coming up next month, I believe, can go into that probably in more detail. Some of the things, I'm not going to steal their thunder, but some of the things they're doing are looking at specific strains of lactic acid bacteria to improve the technology of commercial fermentations, as well as Dr. Johanningsmeyer is looking at lactic acid bacteria making biomolecules that are that have good physiologic effects on humans, like GABA, for example, which is antihypertensive, is produced during fermentations. And so she's looking at those kinds of characteristics, and I'll let her tell you more about that.
All right. Well, we'll look out for her. We'll look out for that presentation. One of the questions that came in from our audience that I thought was an interesting one, because I think we all begin there, and I think many of the people who are here, we have sort of a mix of experts and people who are just interested in fermentation. But a general question that I thought would be an interesting one for you to talk about with regard to food safety was, how do I know if there's something funny going on with my ferment? In other words, what should we just, if you're a home fermenter or somebody who's starting in the business with not a lot of experience, how would you know something was going wrong with your ferment that was a safety issue? Not specifically, let's cast aside all of the pectinolytic stuff and all the other junk, but from a safety point of view, how would I know that something was amiss?
All right, well, first off, it's going to be pretty rare that happens because the sugar is going to be in the vegetables. The lactic acid bacteria will be there, even in very, very small numbers compared to the other microbiota on vegetables. And let me digress with another little story, if I could. If you take 2 cucumbers and you put one on the bench top and you put the other into a beaker of salt water right next to it, and the 2 cucumbers came from the same plant and the same background, and you wait about a week or so and you come back, in the one that's in the glass of salt water, you're going to have a vegetable you can still eat. The one that's on the table is going to be moldy and slimy and very undesirable. So what's going on is this acid production, et cetera, is controlling the microbiota. It's in the water, the same lactic acid bacteria are on both. The same microbiota are on both. But the one in the water, the acids can diffuse all around the fruit and it can touch all the bacteria. Whereas the lactic acid bacteria that are on that cucumber on the table are limited to their tiny little spot. And so, yeah. So the process is pretty much going to go. You'll see turbidity forming, depending on the type of fermentation, and you won't have any horrible smells, things like that. You want to do this anaerobically. You shouldn't see a huge mat of yeast and mold on the surface. Like I said, a small amount is okay. If you really want to take the safe route, my strong advice is to get some pH test paper, which you can order on the web. It's cheap. It costs, you don't need a fancy pH meter. One that'll have a range that goes from like 3 to 5 or something. And, take a little bit of the brine out, dip it in it, and see what the pH is. You want to see a steady drop in pH. And that's the surest way I can tell you, other than people with experience can look at it and know because they've done it so many times. But that's the surest way I can tell you if you're a novice, to make sure things are going right. And as you want to see that steady drop in pH, and you'll see turbidity appearing. That's the cloudiness, is the lactic acid bacteria cells accumulating. It'll become very sour, and you'll smell some acetic acid possibly, and it'll become sour. So, yeah.
So one of the other things that I thought might be interesting for you to comment on was the effect of temperature on your safety curve. So pH versus time, but temperature plays a role in that. And I saw one of your sets of curves that was helpful in that, and you may want to speak a little bit about that, because I know that a lot of folks who are in the production end of this will ferment and then refrigerate in order to slow metabolic activity down in order to deliver a more lively product to the market. So what does refrigeration do to your fermentation safety curves?
Sure. Well, it slows down the acid production rate. Okay, it doesn't stop it. If you do your fermentation in the fridge, it's going to take a long time to get to the kind of quality and flavor that you want. If you do it at room temperature, it could happen very quickly. The speed of fermentation is what's going to change with the temperature. The safety really isn't going to vary much. As long as that fermentation is going on, you're going to be fine. As I said earlier, if the microbial activity is there and the lactic acid bacteria are growing, the Clostridial spores won't germinate. You won't have a problem with botulism. Even if it takes you know, a while for your pH to get down below 4.6, several days, that's fine as long as the pH is steadily dropping and the fermentation is proceeding. So temperature just adjusts that rate, basically. The graph that we had with pH and safety is temperature invariant. It just means that when you're at this pH, you can be assured you've achieved that 5 log reduction in E. coli based on our data. And so it's a pH dependent thing, not a temperature dependent thing. The temperature will give you the time it takes to get there. And for some commercial producers, that's critical. They have to demonstrate that 5 log reduction. So they have to have that pH down. And if they're fermenting up in Michigan in November, it may take a while. Yeah.
Well, Fred, I just got a question in from a participant who asked me, so what did you do about the yeast that I originally had called you about? And so I'll tell you what my answer is. I have battled it tooth and nail, never defeated it. The last time I issued a refund to a customer for a container of pickles that had yeast was yesterday. And we've just managed over the years to evolve our production techniques and our primarily our sanitation methods. And I guess one other thing, which is to encourage the rapid drop of pH in the early stages, the Leuconostoc mesenteroides stage of sauerkraut, as an inhibitor, but not an eliminator of yeast. This is not really a food safety issue, but it does deeply affect the industry, and anybody that's tried to produce commercial quantities of fermented vegetables know that if yeast is an undesirable participant in the process, it can be a very expensive visitor. Fred, do you have any late breaking news on yeast or any things that you think would be interesting to share with the group?
Well, actually, you're not the only one. We get a lot of calls. We get a lot of calls about this issue, particularly with sauerkraut. What I can tell you is there's no easy answer. In some cases, it can come from a particular source of the cabbage because the field or the growing conditions or the transportation conditions, somehow those organisms are more prevalent there than other fields or other trucks that are carrying them or whatever. And so we've certainly seen that. So there's the source is one way to tackle the problem, is to look at where they're coming from. The other is to prevent the organisms from getting into the fermentation in large numbers. And you can do this by various washing procedures, etc. And the sanitation that you mentioned is critical because many of these organisms form very nice biofilms that you can't really even see, particularly in cutting equipment. And so what I've had some folks say they've had great success with throwing away their cutting machine and getting a new one because they tried to sanitize it and it just kept happening. And once they got a new machine, everything stopped. They were having great results again. Yeah. So these organisms are pernicious. They get into stuff. They form biofilms. They're very hard to get rid of when you get them. The other thing is that acetic acid can be inhibitory to yeast. And so that early leuconostoc thing may have something to it in that you're making more acetic acid early on. It can in some cases slow down the yeast growth.
We have a question here. How about non-lactic acid-based ferments such as miso and amino sauce paste? How do you measure safety for those type of ferments? And I'm not familiar with those. I don't know if that's in your wheelhouse, but is that?
Yeah, we don't work a lot with miso and koji and these others, rice and soybean fermentations. Bacillus can play a big role in these, and in some cases it's high pH rather than low pH that's causing the safety factor, in a sense, if you will. But many of these undergo after you. Okay, so there's a couple things that have to happen with soybeans or with rice as a starter material, and that is you have to free up the material to be available, the monomer sugars, etc., amino acids to be available for the lactic acid bacteria and other organisms to ferment. And so often molds are used for this purpose, aspergillus, for example, with koji. And what you have to do, I guess, really is make sure that you have an efficient early process to allow those kinds of breakdowns. It's the same thing with alcohol fermentation. You have to malt barley. The grains have to germinate, and that's actually a process whereby the starches in the seed are broken down into monomer sugars, and this is so the growing plant can basically feed on that. So if you don't have that early initial process successful, the secondary stage of the fermentation with the bacteria is not going to be successful either. I can't give you off the cuff probably a recipe for how to assure safety in every different type of those grain or bean fermentations. But that would be my suggestion as to the key, is to make sure you're efficiently doing the initial stages of the fermentation to make the nutrients that the bacteria need.
Interesting. So kind of dovetailing on to the question prior to that, we had a question here from the audience. What about direct acidification to augment natural fermentation? What potential there to, I could see that it would reinforce pH or reinforce low pH, raising the safety factor.
Yeah, so there's a lot that comes into play with this. It's not a straightforward answer, because what you're doing, it depends on when you add that supplemental acid. If you add it at the beginning, and many large commercial pickle producers, for example, will use recycled brine, which has a lot of lactic acid already in it, but the pH is up above 4 or so before the start of the fermentation, and that gives the lactic acid bacteria plenty of chance to eat the sugar. So the other thing that, other than the pH effects of that, the thing that's going on is buffering, and this is something that we've recently become quite interested in. Particularly with acidified foods and acid foods like salad dressings and things. Buffering is really important for fermentation and for safety of fermentation, because it is how the pH changes with the amount of acid that's produced. And it's not always the same rate. If you have 10 millimolar lactic acid, which is a technical amount, say 0.01% or 5% or something, lactic acid in water, you'll get 1 pH. If you put that same amount into a pickle brine, you'll get a different pH. And it's because there's buffers in that brine that are going to affect how the acid can change pH. And I'm not going to go into specifics because it would take another half hour to really go through that. But we have some new papers out in Journal of Food Science on buffering and buffer modeling. And I have a new research project that we're just starting that's going to be a 5-year project with USDA under our national program staff for food safety. And it's going to focus on how we can use buffer modeling to make a pH graph like the one I showed you that incorporates heterolactic fermentation more efficiently. So we'll be able to tell the ratio of acids, from the ratio of acids, what kind of pH you would get, and how the brine of the fermentation is affecting pH from those acids.
Yeah.
And that's, it's a complicated story, but buffering is important as well as pH. So if you're adding organic acids like acetic acid or lactic acid, you're really influencing that buffering significantly. And you can, if you add too much, you can basically pickle the thing without the brine, without letting the lactic acid bacteria grow. Another even more complicated part of this story is these acids themselves. The organic molecule of the acid itself is inhibitory to the bacteria, aside from pH. There's two effects. And we're able to separate those effects in the laboratory and quantify them. But if you just add hydrochloric acid, for example, which you can get food grade actually, it'll lower the pH, and it's going to make the existing acids more effective in killing. But if you add acetic acid, you're also adding more antimicrobial in addition. So I could go on for a really long time answering that question.
Well, the short answer, I think, would be that yes, you can use non-native acids to that will not stop fermentation. And I think the question was, what about direct acidification to augment? Well, I don't know that it helps fermentation, but if you need to get more acid, if that's what you're after. And speaking of that, we just got a question in about, is there such a thing?
Let me
go ahead.
interrupt you and say one more thing about that. And what you may end up with if you start adding acid like that is an acidified food rather than an acid food. And these are regulated very differently. And there are very different safety factors.
Good point.
So, if you're going to add acids, it should be as a supplement, a minor portion of the acid that's produced. Otherwise, you would be making an acidified food, and you might have to talk to folks at FDA before you try to sell it. And you might have to take a class and thermally process it and things like that. So I'm sorry, go ahead.
We had a question come in. Is there such a thing as over fermented or too tart? If I could just say one thing, I do know from my own commercial experience that yes, there is a thing called too tart, and your customers will tell you when you've achieved that. But Fred, is there any point at which acids either are so sharp or so there's so many hydrogen radicals running around in there that you just can't eat it? Or is there any health or safety consideration there for over fermented foods?
Yeah, I think it'll take the enamel off your teeth.
Yeah.
And that's a problem. But, cabbage has, as I said earlier, about 4% or even greater sugar, 3 to 4%. It's got a lot of sugar. I can show you a graph from some of our earlier papers where research we did in the laboratory, where a cabbage fermentation was left to go for a year, and the acid got up so high because you never run out of sugar, basically. There's always more sugar. The bacteria aren't growing. They can't grow or divide. They're just sitting there slowly metabolizing. And over time, the acid can get up to, several percent where it will be, you have to have a strong constitution to be able to consume it. In terms of other than, I think you have to be nuts to eat it when it gets that sour. I don't know of a particular safety hazard except to your dental work.
Yeah.
But yeah, there's a lot of cucumbers on the other hand. They only have about 2%, and you can ferment them out, and they almost always do. You'll end up with zero sugar. So it's the amount of sugar that's present, and that's actually another. I mean, a key safety point as well is you can ferment all kinds of vegetables. Some vegetables don't have much sugar though, And those are ones that are going to be problematic. Getting that pH down below 4.6 before the sugar runs out is critical. And some kinds of fermentations where you don't have a lot of sugar to begin with, kombucha is a good example. You have to add fruits or sugar to it to allow the lactic acid bacteria to have enough to work on to bring the pH down and make a safe product. So, yeah. So the other way is bad. Too much sugar it's a quality factor, I think. You know, too much acid. I don't know whether I'm stuck or David's stuck. We seem to be. Not sure what's going on. Amelia or Mo, can you hear me? David is stuck.
We can hear you.
Ah, good. Okay, I was worried it was me. So, so hopefully we can get David back online. He seemed to have dissipated momentarily. We'll give him a chance to log back in. I've got your questions here, or a spreadsheet of questions, and I can look them over and see if I can find something till David gets back on. We talked about the role of salt and different vegetables and the nature of temperature. There were several questions again about tempeh and so forth, and we haven't published in this area. I haven't done a lot of work in that area. I'm familiar with the fermentations. If you have a specific question on that, feel free to drop me a line and I can get back to you with a better answer, I think. So, yeah, so let's see, specific bacteria to accelerate pH reduction. Yeah, again, this gets to, so there's a question from Ken I saw pop up on my screen, and in lieu of David being here, I'll answer it. And it was about specific bacteria to accelerate pH reduction. And the answer is the bacteria really are the bugs that you want. There are fermentation starter cultures available. They're often not needed in vegetable fermentations for a long time. And in most commercial fermentations are not initiated with a starter culture. And many, artisan type fermentations aren't either, because the lactic acid bacteria, as I mentioned before, can be in really very small numbers. If you do want to look for a culture that will aid your fermentation, I suggest you attend Dr. Perez-Diaz's talk. She has spent a lot of time working on starter cultures for vegetable fermentations that would give you a rather rapid dominance of lactic acid bacteria, reduce the presence of undesirable bacteria. Because the E. coli in particular, and we've got research papers to show this, some of our recent papers, can grow at the early stages of the fermentation. So the quicker you form acid, the quicker you get the pH down below about 5 or so, which is the limit for the growth of E. coli, the better off you'll be. That doesn't mean you're out of the woods yet in terms of survival of that organism. Yeah, there are cultures available, and you can contact Dr. Perez-Diaz directly. Her contact information is on our website. And so that would be a way to give you a good answer to that question. They can be useful. Oftentimes, they will go pretty well without needing further addition, but they can give you a more uniform and consistent fermentation. Amelia, I see you are on, filling in for David, I guess.
Yes, David lost his internet connection. Sorry about that.
We have hit our time. Our time is up. We wanted to thank Fred, though. Thank you so much for sharing your expertise with us today. I have put Fred's email into our chat box down here. Fred has very graciously offered to answer any additional questions. We got a lot of questions this webinar, and he's happy to take those questions over email. Thank you again, Fred. As he mentioned, our next webinar, it's going to be on Thursday, September 17th, 10 a.m. Pacific, 1 p.m. Eastern Time, with 2 more microbiologists. We have Dr. Perez Diaz and Ben Wolf, the Associate Professor of Biology at Tufts University. They're going to be talking to us about developing new and enhanced pickling techniques and products. So we hope you will join us for that. You can always find links to our webinars on our website, fermentationassociation.org. And thank you again, Fred. Thank you all for joining us today. Any last words, Fred?
Well, it was my pleasure to be here, and I want to say I really appreciate the work the Fermentation Association is doing. It's a wonderful organization. I love interacting with fermenter people, and this is a great venue for us to do tech transfer. And in Linus's talk coming up, you will see some starter culture stuff, I guarantee you.
Great. Thank you so much. And everyone, have a good one.
Goodbye. Thank you.
Bye.